Okay, thank you. Thank Professor Arit for organizing such a very wonderful photothermal webinar series. And I'm really honored to share with the audience of our latest research here. Um, and yeah, so today I will talk about the resolution aspect of photothermal imaging. And before I going into today's topic, I would like to introduce what our lab is doing. Basically, we're a molecular spectroscopy imaging lab where we develop vibrational spectroscopy and related technique for various imaging and microscopy applications. Let me change the pointer. From vibrational dephasing spectroscopy to multimodal coherent Raman imaging. And of course, today's topic, mid-infrared photothermal, MIP microscopy, aka OPTER, right? There are various names of this technique. Okay, so first, if we talk about super resolution, of course, we will immediately think about this fluorescence-based technique that has received this Nobel Prize in 2014. And of course, nothing's perfect. They are relying on this specially fabricated fluorescence molecules. And of course, there are issues including labeling efficiency and photo bleaching. And there, towards this challenge, there are quite a few label-free super resolution methods, and I could only pick a few here from this saturation-based absorption, electronic absorption, and this high-order coherent Raman scattering processes based super resolution technique, and also vibrational infrared spectroscopy-based technique. For example, recently published this bonfire technique, where they use this infrared excitation to and this fluorescence readout to improve the spatial resolution as well. However, of course, especially for the nonlinearity-based super resolution, label-free super resolution technique, they're subject to possible photo damage. And of course, still these are also limited, for example, to graphite or graphene, those specially made molecules or structures. So, of course, we need to develop a universally applicable super resolution imaging, preferably based on absorption. So actually, this is triggered by molecular vibrational spectroscopy, where besides this electronic absorption, there are various of spectroscopy modalities, including Raman scattering and infrared absorption. I think the audience will be very familiar with this. I will skip the details of this. And with the infrared and Raman, we can depict the molecular structure. However, although these two modalities are based on the same type of molecular bond and vibration, they actually have distinctive different selection rules, where Raman transition is based on polarizability change, while infrared absorption or transition will require a dipole moment change. For Raman, I think there are of course, I will skip the time where I was lucky to contribute a few chapters in this stimulated Raman scattering microscopy book, edited by Professor Jixin Chen, Weimin, Yasuyuki Ozeki, and Dario Poli. And on the other hand, this infrared absorption spectroscopy, of course, there are very early attempts for infrared imaging. Well, it didn't catch up because one thing, although it has many orders of magnitudes stronger signal than Raman scattering with complementary information to Raman spectroscopy, but it mostly limited to diffraction limit, where the mid-infrared light has a few micrometer wavelengths that has pretty, I would say, poor spatial resolution. And of course, water absorption and other problems that prevent the broader application of infrared imaging. And this actually triggers us to, and one interesting story is that this is actually one of my early papers based on this non-Raman signal in SRS process, where we try to remove this. For example, here is the pigment in the skin that overwhelms the signal from SRS. And while we and the other groups around the globe that has developed all kinds of techniques to remove the non-Raman's background in SRS, and we actually noticed that this photothermal lensing is actually based on a direct absorption of the pump beam, so that we actually think reversely, that we can, go the other direction, that we can actually amplify this photothermal lensing effect. And that's where we first try to detect this infrared absorption by this photothermal effect, where basically it's an absorption. It start with an absorption. Well, of course, there are more like visible region electronic absorption in very early in history, and then it will have a non-radiative decay where it doesn't emit photon, and eventually the energy will go to the ground and become a change of local temperature. And this temperature, of course, because of thermodynamics law, it will immediately start to dissipate to cooler surroundings, right? Actually, this effect, actually, because of this dNdt is not zero, we in our common life will often, for example, mirage effect, right? We can see it. Actually, this thing is not it's not very new, right? As early as in the 70s, this photothermal detection as an ultra-sensitive spectroscopy technique has been widely used. And recently, including Professor Orit and other groups, has applied this to high sensitivity imaging, up to single molecule sensitivity. And so that I think this question is now obvious, is that we sure can use photothermal detection for molecular spectroscopy. And this is where I, working with Professor Jixin Cheng back in Purdue University, we developed this technique called mid-infrared photothermal photothermal imaging (MIP) microscopy, where we use an visible light to detect the infrared absorption-induced photothermal lensing effect. Where you can see here that when you use a visible light, you can easily differentiate the absorbers here that is smaller than the diffraction limit of the bigger infrared light diffraction limit spot. And we demonstrate the visible resolution, and of course, CD cancer cell imaging. And actually, there are a plethora of other groups. Because of time limit, I couldn't mention all the groups, and I think many of them are already presented their excellent research in this webinar series. And I would briefly mention that besides this photothermal lensing detection, there are other photothermal detection. Actually, photothermal, this term, I would think that it actually is very inclusive. For example, any photons converted to heat that can be described as a general photothermal effect. For example, the photoacoustic approach, where the pulsed absorption that will, of course, lead to a local temperature difference. And then this temperature will lead to an expansion that generates this acoustic wave, right? And of course, recently there are this infrared assisted super resolution photoacoustic technique that has shown very promising results. And also, meanwhile, for example, Professor Idaguchi, right, this quantitative phase imaging based technique to measure the infrared absorption induced optical path length change. And also, not long ago, Professor Garth Simpson has presented his research of fluorescence detected MIP, where they have very nice. They have independent work with Professor Ji Cheng that published in the same issue of this JACS journal. Okay, so I want to mention is that this quantitative phase imaging that can really detect the this molecular spectroscopy, where this mu is the absorption coefficient. By this mu, which is actually the molecular absorption spectroscopy, and then you can actually see the molecular structures, which means it is bond selective. And this actually is in memorial of Professor Gabriel Popescu. Actually, I drove to his lab from Purdue University to UIUC to his lab to learn about this technique. But the challenge is heat dissipation, because as I said, any heat will, of course, immediately start to dissipate to the cooler surroundings. And then to catch this very transient process, you need a very high temporal resolution. However, there are not many fast cameras that can do million or billion frame per second. So this actually relies to a specialized pump probe type of detection scheme, where Harold Edgerton used 10 microsecond flash to freeze the motion of bullets. And similarly, we used an AOM, acousto-optic modulator, to cut the probe beam into bursts of 900 nanosecond pulses that we can, do this transient absorption. Now, this transient heat dissipation. And then we can do this bond selective phase imaging. And this is the actual optical path length decay. Although the decay is pretty slow in this case because it's bulk oil, that the heat dissipation is very slow. But if you zoom into this temperature increase, you will see independent step-like function. That step is because of each single infrared pulse. And you can already see the heat dissipation where the signal drops after each single pulse. And this actually triggers us to think that this thermal decay is so fast that we can actually convert this to, if you time plus time multiply by velocity, then you have a spatial length, right? Physical length, then this is thermal diffusion length. So that we actually model, simulate this in COMSOL, that we can use two beads with the size of 200 nanometers that has a surface distance of 440 nanometers. And we simulated the heat dissipation of these two beads after the initial heat absorption. And then for each pixel, we modulate this heat. We do a Fourier transform of this heat dissipation curve. And then we can see that the center over edge ratio actually increases. This indicates that we can use this chance to further improve the spatial resolution. Actually, when we detect this in a real sample of 200 nanometer beads, we do see an increase of this center over edge ratio. However, as you can see, this trace is not very high in signal noise ratio. That's because we're observing this using an oscilloscope. And of course, there are better instruments than oscilloscope, which is this locking amplifier, especially for digital locking, where they convert this optical, this photocurrent to induce this voltage, and they digitize this and then goes through feeding into this FPGA arrays. And virtually you can extract any harmonic orders without losing any signal noise ratio. And this is actually where we built the first prototype of this high resolution microscope, where we later named this this Pearl microscope. And by imaging this PMMA beads, we can see that if we increase the extraction harmonics of this locking amplifier, we can actually do see an improvement in signal in spatial resolution. However, there's no free lunch. As you can see that when you move into higher orders, the signal level actually decreases. And then eventually we're fighting with the SNR. Okay, so and then another thing is that when we change the infrared excitation power, we do see a linear power dependence. And this is actually very different from the nonlinearity based label-free super resolution technique. And this actually can, is less likely to get photo damage because of this linear power dependence. Okay, then we apply proton imaging to various samples. Of course, here is the mammalian cancer cells, where if we zoom into this small area from the fundamental first harmonic to the eighth harmonic, we can see a very obvious improvement in the spatial resolution, these features, and as well as the water background actually decreased. And of course, we can pinpoint any place of point of interest and measure the infrared spectroscopy by changing the IR wavelength. Yeah, and then of course it has. We also observe other, for example, microtubule structures. Actually, I want to mention that this high order image that suppressed the water background. Actually, actually Professor Jixin Chen in his paper in 2021 has already observed this similar phenomena where he used 21st harmonics with his advanced match filtering technique that he do see a obvious suppression in water background. Another interesting thing is that when we observe this very small model organism, these yeast cells, this is very small, as you can see. And then when we tune to the lipid channel and the protein channels, we actually see that there is a spatial separation between these two droplets. And hold on. And okay, somehow okay, and this if you compare this to conventional SRS microscope, where we observe a bigger yeast cells, and the spatial resolution is very different, and this really give us a lot of opportunities to explore the spatial temporal dynamics of various biological systems. And beyond that, we actually went back to the conventional photothermal imaging, where we use this gold nanoparticles to test the spatial resolution. Actually, when we tune to the higher harmonics with, of course, visible pump pulses, we do see an increase in spatial resolution.
About 3 minutes left, Dr. Zhang. 3 minutes left until the end, if you.
Okay. Yeah. Okay. Thank you. Yeah. And then to summarize, compared to this nonlinearity-based super resolution fluorescence or electronic absorption super resolution technique, where the Pro technique is offers various of advantages in for example, photo damage applicability and spatial resolution. And finally, I would like to use a few 1 minute to cover the biomedical spectroscopy and imaging applications of this MIP or Opter technique, where we focused on this p-tau and amyloid plaques in this Alzheimer's disease brain, where we can observe this beta motif peak difference. And by applying this to brain slices, we can see we can visualize different stages of plaques and the surrounding lipids. And we do see this plaque-specific lipids throughout the whole brain slice. We can see that these lipid dynamics are really not the same. They vary in different AD brain regions. And finally, another technique we're trying to develop is this called simultaneous infrared and Raman imaging, short for INSPIRE. which is we use this general pump probe scheme to use the single probe beam to simultaneously detect the photothermal signal and also the stimulated Raman scattering signal in the same time, so that we can do high content cross-modality imaging of both fingerprint and high wave number region in the same shot. We can this actually give us a lot of opportunities for various studies in, for example, C. elegans. And finally, I would like to thank my students and collaborators and funding sources where I couldn't achieve this without. And thank you very much.
Thank you very much, Professor Zhang, for this very exciting talk and nice results. I wonder if there are any questions. You can ask your questions directly or write in the chat. I don't see. The Marcus Anton, please just speak up.
Hello, thank you for the nice talk. I think you can hear me.
Hi, thank you.
Yes.
I was wondering about the higher harmonics and also the 8th order and 10th order. Do you excite with a sine wave or with a square wave that you can get the higher harmonics?
Hold on, let me see. Which one you're talking about? The excitation?
Yeah, the profile in time of the excitation light. I mean, if you use a single sine wave for excitation, you just have the first order harmonics, because the second order harmonics will give you a 0.
In my opinion, those are pulses, so you have many harmonics, right?
So you
depending on the duration of the pulse, obviously. Is that correct?
Yeah. So unless you're detecting a pure sinusoidal wave, otherwise you will always see some higher harmonics. This actually not new. Actually, for example, musical instruments, right? We can differentiate, for example, piano or violin, because they have different higher harmonics, although they have the base frequency are the same. But right.
Yeah. So then, I think the question was about the excitation mode, and I think your figure here on the slide shows that you have pulses of pump excitation, right? You have short pulses.
Yeah, very short pulses. Yes, exactly.
So you go to very high order of harmonics. If you have infinitely short pulses, you have infinitely many.
Yeah, because the thermal decay is exponential, so it cannot be. It's always different from sinusoidal. And also, if you use short pulses, then the higher, the faster frequency component will show up. If you use like slower, for example, if you use 500 nanosecond, very slow IR pulses, then the heat already conducted to a very far distance, and everything mixes up.
Yes,
okay. Thank you.
Maybe waiting for more questions. I had myself, very general question about the limit. I mean, you mentioned that going to higher harmonics, you lose, of course, signal. I mean, signal to noise ratio worsens. So, what is a good compromise? What is obviously you go much beyond the resolution in terms of infrared wavelengths. How about gaining in the visible range? I mean, what is the resolution limit that you envision with this technique?
Yeah, that's a very good question. We used this green and blue probe light, and so it actually varies for of course different samples will have a little bit different responses because of the absorption cross section and heat conductivity properties. And in general, we can usually improve basically it's double the resolving power. Yeah, usually.
Yeah. So, but I suppose this resolution or resolution power, or the resolution limit will depend on the vibrational band that you look at. I mean, it will not be the same looking at lipids or proteins or other organelles, right? This definitely will depend on which vibrational mode you are probing.
Yeah, it depends on the.
But
sure,
can you give us some idea of the limit, right? I mean, what is the brightest signal you get, and what is then the smallest structure with your technique and equipment that you can reach? So typically, is that what is on this graph? I mean, in tens of nanometers, or that's dead. Okay. So you are sub wavelength, right?
Yes,
optical
of course.
wavelength.
It's yeah. Sure. So yeah, we basically the most popular bands in the infrared region is, for example, the 1750 CO double band, right?
Yeah.
And also the IMAD band, 1650 and 1550 bands, yeah. And these structures we can for sure visualize. And the typical structures as smallest we see in the, for example, the yeast cells we visualize is 170 nanometers.
So well below the visible wavelength.
Yes. And this, yeah, of course. So, and one thing is that these structures, for example, lipid droplets, it's actually not easy to be labeled by fluorescence probes, because fluorescence molecules are pretty bulky. So this is actually another advantage of this MIP or Pearl or general opter imaging technique. Yeah.
All right. I don't see any more questions. We still have a few minutes, so don't hesitate to ask.
Sorry. There are two questions from René and
okay.
Yeah.
So please go ahead. I don't see them. Yeah, go ahead, please.
Oh, okay. Sorry. Professor Zhang, thank you for the great talk. I also was wondering about this higher order process, and you showed the linear power dependence sort of here in the previous slide. And you mentioned you thought this would be advantageous for mitigating photo damage. Can you just comment on that some more?
Yes.
Yeah, so that's a very good question. Yeah, because for example, the saturated fluorescence technique, right? If you are in a linear region, then your detected signal would be linear with the excitation power, right? But in this case, then they use pure sinusoidal, so then there's no harmonics. And then unless they use very high power, then it drives the signal out of the linear region, then it will show some higher harmonics. Then if you extract these harmonics, you reach the super resolution technique, super resolution region, right? So that nonlinearity will require higher amount of power. So in other words, for example, if it's a quadratic dependence, then if you drop the pump power, for example, 10 times, then your signal will drop 100 times, right? That will be a loss. So in other words, for nonlinearity based, they will need higher peak power, and this higher peak power will increase the chance of photo damage. Okay. And in this case, if you have linear power dependence, then you can use a lower this excitation energy and still have a reasonably good signal.
Okay,
if
that makes sense.
Answer the question. Yeah. Thanks.
Yeah. There is another question by Frank Zichos.
Yeah, thank you very much also for the very nice talk. My question is going along the same direction. So actually, isn't the use of the pulse at the end not really favorable? Would it not be nicer to change the modulation frequency with a sign that you can put everything into one mode just and measure at a higher frequency, because you're just scanning over the thermal conductivity or the frequency dependence of the thermal conductivity? And the second question was also related. So how is this? I mean, the resolution will be related to the thermal conductivity of your medium at the end. I mean, in this approach, and maybe also at the heterogeneity of the medium. So it will spatially depend on how the thermal conductivity is. Of course, in the cells, mostly this is maybe water, but
yeah.
sense?
Yes. Yeah. So of course, actually, this would. Do you mind if I answer the second question first? You were talking about the surrounding environments, right?
Yeah.
So yeah, we are mostly looking at the cells and tissues, and they are mostly in water environment. So that's the biggest heat sink. Yeah. And if you
some maybe
yeah, and
to substrate and things like that, where the effective thermal conductivity changes, for example.
Yeah, but then of course you can always extract the diffraction limited size from the fundamental frequency, which is conventional MIP imaging, and also for bigger particles, bigger than the diffraction limit of the probe beam, then only the edge matters. Yeah, so then if you can detect the edge in high resolution, then you can have the higher resolution for the whole image. And I kind of missed your first question. I'm sorry. What was that?
I mean, it was related to the pulse. So if you create a pulse, you create all the harmonics, and then you essentially use one of the harmonics to do something, maybe a higher harmonics, which has only a fraction of the intensity as the fundamental one. You could also directly go to a sinusoidal pump modulation at this frequency and measure with the same spatial resolution, wouldn't you? I mean, and then you don't.
Yeah. So that's why this digital locking is very important, because you use the same this photodiode. It digitizes everything. Then you can simultaneously get from the first harmonic to any higher orders limited by the signal noise ratio.
Oh, okay, no, my question was related otherwise, but most of the infrared lasers anyway come in a pulsed mode, and then you don't have the chance to really do a fundamental.
Exactly. Yeah. Yeah. Sure. Because the heat dissipation. If you use sinusoidal, then it will just blur everything up.
All right. Maybe we have still time for last short question by Yako Ton.
Hi.
Can you hear me?
Yes.
Go ahead. Yes.
My question is, when using this digital lock-in that you call it, is it then possible to real times look at your cells?
Yeah. Exactly. That's what we do. Yeah.
Okay.